This is episode 86 of the Immunology Podcast, Myeloid Cells with Dr. Kipp Weiskopf. Hey everyone, this is Dr. Jason Goldsmith and Dr. Brenda Rout. Welcome back to the Immunology Podcast. We have conversations with immunologists. The Immunology Podcast is brought to you by STEMCELL Technologies, a global biotechnology company supporting life science research and fostering communication and collaboration in science. If you enjoy the Immunology Podcast, please rate us and leave us a review. We're always looking for feedback on how the podcast can be improved and for suggestions on guests. Today, we have Dr. Kipp Weiskopf from the Whitehead Institute and Dana-Farber Institute on the podcast to talk about his research on increasing myeloid cells' ability to target cancer cells. We've also got our usual roundup of recent highlights in immunology news coming up. But first...
Are you studying immune cells? I would take that if you're listening to the podcast, that's probably the case. Well, if you want, you can get a free infographic wall chart that provides an overview of key cytokines for expansion, differentiation, and characterization of major immune cell types. To learn more, visit stemcell.com/cytokines-wall chart. All your reference in one wall chart. How cool is that?
That's awesome. I'm thinking about getting some for my lab, actually.
Hey, that's not too bad, right?
We need some stuff on the walls. It's all like just white.
Oh no, that's such a sad lab. You need to have, the cytokine wall chart, the cells in the blood wall chart, maybe a fluorochrome wall chart for your cytometry assays.
Exactly.
That's how it is.
All right, Brenda. Well, summer is almost over.
Shut up. I don't la 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 la.
Wait.
I'm not.
Does it actually change for you at all? It just means it gets dark at night, but you're still inside in fluorescent off lab every day, right? Is it different for you?
No, my heart is getting empty anyways by this lack of sun and fluorescent light. Well, I mean, it's still a month to the solstice, the equinox, sorry. So I would, I'm gonna just ignore the fact that summer is on its way out. I'm just gonna pretend it's not happening.
My kids go back to school soon, and that is a wonderful thing.
Finally, some peace and quiet around the house.
I mean, it's just like every week it's a different camp schedule and shuttling them off. And then sometimes they're off camp and they bring their phones over and friends over and their house rattles. The friends' loudness. It's quite impressive.
I mean, kids are loud. I don't know what you expected.
I expect them to go to school during the day.
Well,
small thing.
Don't worry. They will take them off your hands very soon. Then you will miss them. You'll be,
I will miss them, but I see them every day. It's quiet. All right. Well, we need to hop into this. You want resident memory or TCR receptors first? You pick.
Ooh. Let's go for the memory. For the resident memory.
Memories. All right. Here we go.
Blow my mind.
In Nature, published the 14th of August, Human Organoids from the autologous tissue-resident immune compartment. First author is Timothy Ricaldin. Last three authors who are all corresponding, Lorraine Cabon, J. Gray Camp, and Nikolchi Gajorevsky. Sorry for all the dead names I've left on the field of pronunciation there. So we can grow organoids in tissue. We can take stem cells and make pluripotent from iPSC them, so induce pluripotent stem cells and grow up intestinal organoids, or you can take just intestinal stem cells and with less work, just grow them up. But you know what they don't have? They don't have the immune cells. And co-culturing them has never really worked before in setting things up. There's been a lot of technical hurdles. But this group figured out how to do it. So what they do is they take from a tissue sample from a person, they take the epithelial cells, which you know how to do, and they make an organoid out of that. But they then do a cytokine-free, scaffold-based tissue resident memory isolation. And so this is a process where there's no chemicals that chew it up to get them out, which causes the memory cells apparently to go haywire. But they put a element in that's a scaffolding element that they're attracted to, and then they can make sure by flow that they're the proper cell. Right? And then they throw these into culture. And the first thing they do is they see what happens. And the resident memory cells intercalate into the epithelial cells.
Just like that.
Uh-huh. It's like they know what to do.
Oh, wow.
So that's really cool. All right. So then they show this as the case. And then I think this is, if you're in the field, you should go read this paper for all the interesting single cell RNA-seq and determining what happens in the different populations, and if you add them in or don't add them in, and how the resident memory cell shifts. And they have program in the absence of epithelial cells, they actually have more programs to like try to find them, the right compartment. And then when it's there, it actually shuts down because it's found them. And so that doesn't need to migrate anymore. It's more about cell-cell interactions and everything else. But cool, they're resident memory cells. They look at the T cell epitopes and then they compare these to PMBCs, which are added in, and compare all of that, right? So they use the PMBCs from someone as a control, so on and so forth. Okay, so that's really cool. They understand the transcriptomics and everything else. They then look at immune checkpoint colitis systems, right? So what they really want to understand is like, you want to be able to do something with this, right? You don't want to just be able to, okay, you can do it, great. But they wanted to be able to like, well, what is this system for? And so they were able to use it to interrogate checkpoint colitis. And they show that in inflamed tissue being brought in, it recapitulates, but they can also induce it. And that when they induce it, they use solitomab, which is a bispecific T cell engager that induces intestinal toxicity, and it's known in people, and it was a problem in the drug. So they induce the intestinal toxicity, and they see the resident memory cells activate, causing, as compared to being co-cultured with PMBCs, it's the resident memory cells that are driving the interaction. So we had a bispecific that's supposed to be an immune checkpoint, overcoming, a cancer drug, but it had too much toxicity, right? A bispecific T cell engager, but it had too much gut tox to go into market. And then in this system, they can see that with this drug, it's engaging the resident memory cells and driving immune activation and damaging the epithelia. But if you put in PMBCs, it doesn't do anything. So it's actually engaging the local resident memory cell and causing tissue damage. It's recapitulating the human disease. And so that's kind of where they end this. They describe some mechanism, and they look at what happens with cytotoxic CD8 IELs and other things in the system, and what happens to the PMBCs that are added to it. And yes, if you add PMBCs, you have synergy with the T resident memory cells, right? They're going to direct the traffic. But if you just put the PMBCs in, nothing happens.
Sorry, do you mean PBMCs?
Sorry, PBMCs. Yeah, it's a lot.
Okay, sorry. It was hurting my ears.
Yeah, PBMCs. So you put the PBMCs in, and then you get what you need, right? So that it'll synergize. But you have to have the resident cells to have any effect from this drug at all.
Right. That's cool, because it's something you could only would have done if you had this organized system, which you had the resident cells included as resident cells, and not just as a PBMCs on the side.
And then they were able to drive it with anti-CTLA4 and anti-PD-1 antibodies.
Right. Because that is, just to say, because colitis is a common side effect of...
Right. It's not as robust as this cross-linking bispecific, but they get it to work.
Mm-hmm. Mm-hmm.
And so that's really cool.
Yeah, for sure. And when I look into like the markers of memory residency, they're all kind of still check out. Like, I don't know, CD69, CD103, Hobbit. I don't know. Those are like typical markers of these cells.
Yeah. So they measure every marker on there. So they have CD38. They see expansion with Ki67. This paper is full of single cell RNA-seq and markers all over the place, which helps prove the story, but isn't the cool part of the story to like share with audience.
Right. All right. Okay. Very nice. Thanks. As always, thanks for sharing. All right. I'm gonna move on with a pretty cool, I think very nice story on regulatory T cells. And a little glimpse on how Tregs work, what is kind of the under the hood happening with FOXP3 and all its friends that are required for Tregs to maintain their identity and their suppressive function in a way. So I think this is a very cool story to illustrate the complexity of maintaining a Treg phenotype that goes beyond FOXP3. So the paper is called Transcription Factor IKZF1, aka Icarus, associates with FOXP3 to repress gene expression in Treg cells and limit autoimmunity and anti-tumor immunity. And it was published in Immunity, 6th of August. And the first author is Kenji Ichiyama from the very well-known Treg lab of Shimon Sakaguchi at Osaka University. So, this is one of the first labs. Shimon Sakaguchi is um, really was one of the pioneers in understanding the function of Tregs and suppressor cells. So it's nice to see something coming from his lab. And the most important kind of the basics of this of the study is the understanding that besides besides FOXP3, there are a bunch of other transcription factor epigenetic modifiers that are proteins that together with FOXP3 carry out the task required for a CD4 T cell to become uh to sustain to stay and to as a Treg and to do this function, right? So when you think about a regulatory T cell, you want a cell that can not only inhibit the activation of other T cells or the function of other T cells, and there are many ways through which regulatory T cells can do this. It's it's still kind of a debate of which are the most important. They're probably kind of vary in different circumstances, require different mechanisms. So you have, the high CD25 expression, which is the IL-2 receptor. So you're collecting IL-2 from the media or from the environment and sequestering it away from conventional cells. You have certain suppression programs that require close contact, like CTLA4 binding to CD28 or things like that. And there is CTLA4 binding to B7 or so CD886 in antigen presenting cells, production of metabolites that are suppressive. But there's also another thing is that regulatory T cells, they have to be able to withstand activation and withstand certain level of inflammatory conditions and stay Tregs. Because when you think about it, they are being activated on their TCR, they are being exposed to inflammatory cytokines. So a lot of also of what makes a regulatory Treg a Treg is the absence of certain pro-inflammatory cytokine production, for example, particularly, and I think this is important for this paper, is Tregs should not produce interferon gamma, which is highly common on activated T cells, and Tregs should not produce IL-2. And this is part of like kind of the definition of a functional regulatory T cell. So there's a family of transcription factors known as the Icarus family, which are zinc finger transcription factors that control various types of gene expression, including some types of chromatin remodeling, and they are kind of expressed throughout hematopoietic cells, and they're very important in hematopoiesis. That has been known for a while. They're also important for B cell differentiation, and they're also important for T cell differentiation. So there's 5 members. I like to just gonna quickly mention them. So basically, the genes are called IKZF1 to 5, and the common names Icaros, Helios, Aiolos, Eos, and Pegasus. So all these nice Greek names. And one of the things that the first 4 are also expressed in regulatory T cells. And maybe those, regulatory T cell aficionados might have also recognized Helios. Helios is nowadays considered a marker for stable kind of Treg function. And we know that disrupting Helios or the loss of Helios activity is related to loss of kind of Treg identity as well. So nowadays, FOXP3 alone, particularly on human Tregs, is not sufficient to properly identify as regulatory T cells with a regulatory phenotype. Also because human T cells will express FOXP3 also in conventional cells. So they wanted to look closer into the function of the first member of the family, Icarus, in Treg identity. So what they did is that they started kind of thinking of the functional domains of this protein. So I mentioned that these are zinc fingers, and so there's the Icarus has several zinc finger motifs distributed along the protein, and what they did is to try to find which of these zinc fingers is associated with Icarus known association to FOXP3. So proteins like Icarus and other members of this family will make complexes with FOXP3 and other epigenetic modificators and expression and molecule and proteins that are regulating gene expression, and they are going to be part of the complexes that guide the. If you think of FOXP3 as kind of a conductor, FOXP3 is a transcription factor as well. And the expression of FOXP3 in T cells results in the inhibition, the suppression of certain genes, and on the increased expression of other genes. And this is what in the end gives Tregs their signature. But although FOXP3 is necessary, it's not sufficient. FOXP3 works together with other transcription factors and modulators, such as the Icarus family. So when they know that FOXP3 and Icarus, they interact some way, and they show that there's one of the exons of Icarus, so they kind of knock out one exon at a time, basically, and they show that it is the zinc finger motifs that are located in, or the domain that is located in the exon 5 that is required in order for Icarus to interact with FOXP3. So if you eliminate that domain, Icarus still exists, but it doesn't associate with FOXP3 anymore. And this is kind of the starting. This is the basics of their experiments. So they have mice in which they have a Treg specific deletion of exon 5 in Icarus. So basically, exon 5 floxed, then crossed with a Cree under FOXP3 expression. So this is very, very straightforward. And basically, these mice develop autoimmunity comparable to kind of a scurvy mice, kind of complete autoimmunity that is lethal. And if you let the mice, usually they don't make it to 2 months, and they have massive inflammation. They have reduced body weight, lymphadenopathy, splenomegaly. So all these kind of enlarged spleen, infiltration of leukocytes in skin. So this is very similar to the scurvy phenotype. So clearly that interaction is very important for the kind of function of regulatory T cells. So what they show is that, kind of long story short, the main effect that they see is that cells or these mice or the regulatory T cells that lack this binding, this interaction between Icarus and FOXP3, the most salient effect is that they start producing interferon gamma. So or better said, they stop repressing the expression of interferon gamma in the Tregs. And this has this is really a major problem for regulatory T cells that start expressing interferon gamma, and they become kind of this known fragile, unstable Tregs. And these cells they cease to be suppressive. So they're not capable of doing their job because mostly they're producing interferon gamma. And so they show that a lot of these, if you have in vitro, the stability of these Tregs is, if you block interferon gamma, then you can in some ways contain this loss of Treg identity. So it really is a lot of it is mediated by production of interferon gamma by uh, Icarus exon 5 knockout T cells. They do a lot of analysis in the where they look about on which are the genes that have been modified. So there's of course a whole change in the signature of the Tregs, but basically they show that it is through changes in these repression of these genes that really affects the functionality of the Tregs. And in the interesting thing is that you can have so they think, well, where can we use this? So this fragile Tregs also they're less suppressive in the context of a tumor. So potentially they do some mice, some B16 melanoma experiments, and they show that this knockout can generate Tregs that are no longer um, sustaining tumor growth. And basically, if you could maybe target these Icarus proteins, you could potentially increase, for example, anti-tumor immunity. And they do some tests with that. They show that there's a inhibitor that can degrade Icarus and I think it's Iolos. So there's another member of this Icarus family, and that this potentially could be used for targeting Tregs in a tumor context.
Did you say YOLO?
No, Iolos.
YOLO. All right, I was like, wow, you went from Icarus to YOLO. Oh,
YOLO.
Interesting. So this is a highly all about transcriptional regulation.
Yeah.
It's very interesting basic biology. I wonder if they're going to try to, they've never done a good job making transcriptional drug products, but maybe with, more gene approaches or CAR Ts or what have you, this would be super valuable, right? Because you can then push the environment.
Yeah. I mean, it's really interesting to understand how this repression and all the mechanisms by which this, because you have FOXP3 and then you end up having, genes that are suppressed, genes that are expressed differently. And it's not only FOXP3, it's all of these other proteins that make all these complexes. And then if you don't have FOXP3, you don't have Tregs, clearly. But then if you don't have all these other epigenetic regulators or transcription factors, then you don't have like parts, because other parts of this Tregs, they've produced interferon gamma, for example, but there are no changes in other Treg genes like CTLA4, for example. They still make the same amount of CTLA4, or there's other molecules that don't change, but it's particularly the repression of these inflammatory cytokines seems to be mediated by this complex in particular.
Yeah, interesting. All right. Well, keeping on more T cells.
Sure.
It's also in nature. Received the 16th of July, 23, except to the 7th of August of 24.
Oof.
Structure of a fully assembled gamma delta T cell antigen receptor. Benjamin S. Gully is the first author. Last author is Simone J. Davis. Oh, and Joao Ferreira Fernandez, Sachit Guna CJ, Mai Vong are all equal first authors with Benjamin Gully. And then other corresponding author is Jamie Rossjohn. All right. High level. We know the structure of the alpha beta T cell receptor with cryo-electron microscopy. It's pretty rigid. You see a really cool molecular dock between it and MHC. You see its tail that's membrane bound, surrounded by the various CD3 epitopes, right? The subunits of CD3. And the structure is pretty well described. We're like, well, alpha beta TCRs are very antigen specific, locked in tight. That's it. That's what you have going on. Gamma delta are a little more promiscuous, right? They use MHC-like binding. They will bind to receptors that will accept, they'll bind to immune complexes that accept a few different ligands, not just this one thing from an MHC, right? So they're a little more permissive. And they went and did a structure. And this paper has lots of cool X-ray, 3D protein structures and X-ray crystallography. But most importantly, the head of the TCR is a wee bit more floppy. They couldn't get full resolution of it. Probably because it's so promiscuous in what it binds to. It binds to things in a bunch of different ways. And we put antigen in to the mix. And that's the big take home from this. You see similar 3D conformation of the inserting membrane with the gamma delta T cell, or the gamma delta to the alpha beta with the CD3 units, but there's less that holds it in there. So instead of it being like a Y that just sits out and stuck, the Y kind of moves within the pocket, and the arms of the Y move a little bit more. So it's a little just more flexible, right? And so then they swap parts of it of the protein out, and they remove the flexibility. They see that the alpha beta require less antigen as well to saturate the receptor and induce a response. So they see that the gamma delta has a higher KD, so to speak, than an alpha beta to get the same level of response. And so they do some studies with that. They go and use FRET to look at associations, and FLIM-FRET really to measure the ability to understand if the distance is different between the two when they bind. And it's not so much of in the floppiness makes the density, shows that the distance changes, right? So if you're more flexible, you have more variation in distance on binding. And so you're going to have, if you set up the FLIM-FRET experiment, right, which measures distance by light being released between an antigen and a receptor, and it's really tight, that's going to be a small number with lots of FRET. And it's floppier, and the FRET efficiency goes down in this floppier environment. So that's an orthogonal way that they show this. And that's basically the paper. But they have a structure. They're really getting into the protein mechanism of how gamma delta T cells are different. And it provides some interesting information, which is, we were talking, even at the conference, right, about how once you know the structures of things, then AI can start doing cool stuff. And this was a big key unlock because at this point it had been a mystery. Now, the fact that it's flexible makes it can be harder to predict, but also knowing that it can be flexible has value. And there you go.
All right.
Delta T cells flop around in the breeze.
floppy T cells. So here it is a V delta 3 gamma 8 CCR. Do they expect, because there are different flavors, right, of the gamma delta cells that seem to have.
They pick a specific V combination that works really well. And they go into like why they pick one and not the other, and it mostly has to do with how well they know of a very specific ligand for it.
What is the ligand for this TCR that they're looking to?
With MR1.
Okay. All right. I mean, it's super nice. I'm surprised that it took that only now. I kind of thought we already knew this. I guess not.
We knew it was a little more promiscuous, but we never had a structure because you have to use cryo-EM, and that requires pure protein and then lipid raft scaffolding, and they used antibodies to hold it in place.
Right.
And there's a bunch of technology here if you really want to get into the weeds of why it took a while, but in the end, it's floppy.
It's floppy. All right. Well, floppy T cells, floppy TCRs. That's why, you know what? I'm not going to make that joke. I'm going to move on to the last paper of the day. Not sure. I don't have a good segue. I'm going to talk about autoimmune organ damage and why ILC1s are responsible for it in certain cases. So the paper is called Amplification of Autoimmune Organ Damage by NKp46 Activated ILC1s, also published in Nature. We have a Nature-heavy roundup today. On August 5, this is a accelerated preview. First authors are, let's see if I do it right, Stylianos Yasom Viniaris Georgalis, Tom Ashman, and Katerina Stergiola, I guess. I hope I did not completely butcher this. And then from the corresponding authors are Antigoni Triantafilopoulos, Andreas Diefenbach, and Matoshi Kanda. These are the mostly at Charité University, medical university in Berlin. And basically, in this paper, they're looking into kind of kidney injury associated with lupus or what is called as lupus nephritis. And they kind of want to, always the question is that when you have patients with lupus, usually all these patients have autoantibodies, but not all of them develop severe inflammatory disease. And the kidney is a particular organ that is very much affected in lupus. Um, and so they want to try to understand what are the other triggers on top of having autoantibodies that are responsible for the damage that happens in organs, particularly in kidneys. So they start working with some mouse models that mimic the lupus condition. Or basically, they give mice lupus. They have these NZBWF1 mice, which develop a type 1 interferon driven disease that is similar to systemic lupus in humans. And which is if you administer poly-IC to the mice, this synchronizes the onset of disease, and you have this nice cohort of mice that are starting getting disease. But you can block the disease with anti-interferon alpha receptor antibodies. So basically, this is kind of the model that they use. So one of the things that they're focused on is ILC1, so innate lymphocyte cells, which are very much present in tissues. And they're known for being kind of rheostat from responding to inflammation and affecting tissue homeostasis around them. So they are usually important players when it comes to tissue homeostasis. So one of the things that they see is that when they give the mice lupus, one of the receptors that really pops up is this receptor called NKP46, which is an activating receptor that is, originally found in K cells, which are innate lymphocytes, and basically activation of the receptor activates the cells and induces usually a pro-inflammatory or some kind of activation program. So when they look into, they show that in lupus nephritis, in kind of inflamed kidneys, this receptor is upregulated in ILCs, and that blocking the function of this receptor with, for example, blocking antibodies improves the parameters of kidney damage, and without affecting kind of, for example, the serum, the titers of the kind of the self antibodies, which are the hallmark of lupus, but they do kind of seem to affect downstream of that, improve the nephritis. There's a lot of kind of measurements that they do. The loss of podocytes, which are the cells in the kidney, the injury in the capillary endothelial, and they do a lot of kind of more detailed analyses. I don't want to go too long, but they show that there are certain specific subsets of innate lymphocytes that are expressing this receptor, and that they are expressing other receptors such as CD137, and they respond to. They don't know exactly how they're being activated, how this receptor is being, what is the ligand of this receptor, but they do express a lot of it, and blocking it ameliorates the inflammation, basically. So downstream of these cells, what they show is that, and particularly, is the production of CFS2 that seems to mediate this kind of inflammation downstream. It's not the cells themselves that are inducing the inflammation. They are not the ones that are, damaging the tissue, but they are acting as mediators through the production of CFS2, which in hand induces the differentiation of monocytes into pro-inflammatory macrophages that are in the tissue. And that is kind of the connection that they see between these ILCs, the expression of this receptor, and downstream, the activation of macrophages that are actually inflammatory, and they're damaging the tissue themselves. So basically what they show is that there is this events that happen, and this connection between resident ILCs, which we know often do this. They often act as intermediaries in a way and activate other more effector cells. And they show that they can induce the differentiation of monocytes, and partly monocytes from the blood, to carry on the nephritis and the damage of the kidney tissue, basically. And this expansion of these macrophages is detrimental. They also talk about a different type of macrophages that are anti-inflammatory. I have to admit, it's not completely clear, because it looks like they can also differentiate into macrophages that are less pro-inflammatory. But it's not completely clear for me how is the balance mediated. But in general, activation through NKp46 has a negative impact on the nephritis. And they also show some human data. They also show that there's some of these transcripts in human nephritis as well. So they think that it's probably also all these experiments are in mice, but they probably can translate to some extent in human. And as you were mentioning from the previous from your first paper, this paper was in revision for 2 years. So sometimes you just need. I'm not sure what took so long, but yeah, that was a long roller coaster for the authors, I'm sure.
It was reviewer number 3.
Yeah.
Well, you just talked about macrophages. And we're going to get a giant dose of the importance of myeloid cells in checkpoint therapy and how to get around myeloid suppression when we talk to Dr. Kipp Weiskopf at the Whitehead Institute in just a moment. But what if you need more monocytes and macrophages in your life of some form? Do you want to differentiate some human pluripotent stem cells into monocytes? Well, you should check out the STEM-Diff monocyte kit from STEMCELL Technologies. It generates millions of monocytes that are ready for downstream assays or for further development to macrophages or dendritic cells. Learn more at www.stemcell.com/monocyte-kit.
For a second part of our episode today, we are joined by Dr. Kipp Weiskopf. He is Valhalla Whitehead Fellow at the Whitehead Institute. And I'm very excited about today's talk because a lot of his work has involved a particular molecular interaction and these not don't eat me signals, which I think is very interesting and has a lot of potential in oncology. So I want to let him talk more about it, but I'm very excited about today's conversation. Dr. Weiskopf, thank you so much for joining us today for the Immunology Podcast.
Thank you so much, Brenda and Jason, for having me on the show.
Thank you for being here. All right. So first off, before we get into macrophages and don't meet me, what is a Valhalla Fellow? Do you like I have to ask because of the name? Just off the top, because
Yes.
It sounds very epic. But a Whitehead Fellow is an independent faculty fellow position. So it's a position intended for people to start their own labs in lieu of a conventional postdoc. And so people straight out of graduate school who are interested in kind of having an independent career early on. It's an independent faculty fellow position where we're given our own space, we're given a startup package, and we're given principal investigator status to apply for federal grants or other philanthropic grants. A Valhalla Whitehead Fellow is supported by the Valhalla Institute, and it's a named fellowship here at our institute based on our funding sources who have been incredible in supporting us. So we're incredibly thankful to the Valhalla Foundation for everything that they do for us.
I'm kind of disappointed that it's not the Nordic gods funding your chair. That's okay. That's okay. I think we'll have to do.
For a science researcher, any source of funding feels like a gift from the gods, right? So we definitely appreciate it.
Since it's a foundation, secretly Odin is funding it, and it's just how it works, right?
You never know.
That's right. It must be. Yeah. It doesn't come with a giant hammer or anything, but maybe someday down the line.
All
right. When you're strong enough to hold it. But so this position you acquired right after you finished your MD PhD. Is that how the transition worked?
Yeah, so it's a non-conventional position, I'd say, and I took a non-conventional route to get here also. So I was an MD PhD student out at Stanford. I did my PhD in Irv Weissman's lab out there, which is a big stem cell biology, immunology, cancer biology lab. And after I finished up the full training, the MD and the PhD, I decided to kind of train and become licensed as a physician. So I came from Stanford to the Boston area, completed my training in internal medicine at Brigham and Women's Hospital to complete residency training, and then joined the Hematology and Oncology Fellowship Program at Dana-Farber, a clinical fellowship program, and completed kind of the bulk of my clinical requirements before starting as a Whitehead Fellow here at the Whitehead Institute back in October of 2019 to start my own group, start my own lab, and continue some of the work we were doing on understanding the interactions between the immune system and cancer. And so most people who start in these positions typically come directly from graduate school to kind of lead their own research effort. My path was a little bit more circuitous, where I went and completed or have done clinical training before coming back to the research world.
All right. Very impressive. Very, very interesting. Thanks for sharing your path. I'm sure many listeners are going to be very interested in that. Okay. Jason, do you have any questions? And can we start with the
Oh, yeah,
science?
let's start with macrophage here. So macrophages are hungry. They like to eat things. I guess before we get into how it all goes wrong, do you want to provide a little basic information on like how it goes right so that we can understand when things break better in cancer?
Sure. Yeah. So I think macrophages are the most interesting cells in the body. I think so for a number of different reasons. But number one, if you look at them just based on sheer cell mass, they're the most common immune cell, and they're found in tissues scattered all throughout the body as resident macrophages. So it's estimated that the average person has about 1 pound of macrophages in their body, which I think is pretty incredible. Number two, they're endowed with this special ability to perform the process of phagocytosis, which is a really cool biological process and a way to eliminate pathogens or foreign material debris, where macrophages are incredibly gifted at engulfing and destroying this foreign material and eliminating it from the body. And then they're essentially involved in all states of human health and disease, everything from wound healing to responding to infections, to metabolic syndromes, to atherosclerosis, to neurodegeneration, autoimmunity. Everything involves macrophages because they're everywhere. And they have a particularly complex relationship with cancer, where it's been found time and time again in multiple different models, solid tumors and hematologic malignancies, that often when you have more macrophages infiltrating into the tumor microenvironment, this typically corresponds with worse patient outcomes, worse patient prognosis, worse overall survival. So the general thinking in the field is that macrophages in tumors are bad actors, that they're doing bad things at baseline, that they're promoting the growth of cancer cells by secreting growth factors. They're secreting things like VEGF to promote angiogenesis. They're remodeling the extracellular matrix that causes cancer cells to invade. And all of these things lead to the growth and dissemination of cancer to make cancer more aggressive.
I just want to say I am very happy that you're giving, myeloid cells their due, because I don't think we do that enough in this show. So I thank you for that. And so a question I was asked, like when I hear about the negative impact of macrophages in tumors, there are situations which that is a good thing, like this pathways or these activities that are detrimental to tumor clearance are actually what the macrophages are meant to do, right? Like in tissue repair. So what are the pathways, or what are the functions of macrophages that are usually found in like good situations that then are really uh, being particularly problematic in a tumor?
That's a really good question. So people often talk about this concept of macrophage polarization, right? This concept of M1 versus M2 polarization, where the M1 macrophages are those that are classically activated or pro-inflammatory macrophages that are responding to things like infections. So things that are super inflammatory to try to clear out any bad material that might be present. The M2 macrophages are believed to be these alternative activated macrophages that are more immunosuppressive, that exist in the tumor microenvironment, and may be more like macrophages that are responding at the end of an infection to clear out or remove any debris that might be present and promote a wound healing response. And so the general thinking is that macrophages in the tumor microenvironment kind of see the tumor as a chronic wound that's just never healing appropriately. And they're there because they're responding correctly to the stimuli that they're receiving and doing everything they can to try to heal that wound. But in doing so, they're secreting lots of growth factors. They're secreting things like EGFR ligands, and they're secreting immunosuppressive cytokines like TGF-beta or IL-10 in some contexts. And that these things generally allow the tumor to be more aggressive and suppress immune responses by the adaptive immune system to prevent the adaptive immune system from recognizing cancer cells as foreign and attacking those cancer cells. And so that's kind of this general paradigm that's been proposed. Where things get much more complicated with macrophages and myeloid cells is that everybody kind of acknowledges that at the functional level, these two states are probably true, that there are some macrophages or myeloid cells that can attack the tumors. There's some macrophages and myeloid cells that can promote the growth of tumors or suppress the tumor microenvironment. But definitely this very simplistic paradox of M1 versus M2, everybody kind of agrees doesn't actually exist at the molecular level. Everybody agrees that there are many more potential activation states or polarization states of macrophages that can exist. And what a lot of the single cell RNA sequencing experiments have shown is that, there are potentially many different stimuli that are activating macrophages in the tumor microenvironment. And so there definitely isn't kind of this simplistic concept of M1 versus M2, but I like to think of it in that way from a functional perspective, just because whatever macrophage activation states might exist, there are certain stimuli that we think can, consistently and uniformly act on different macrophage populations that are there to mount them or provoke them to be more effective against cancer.
So you've described how wound healing macrophage M2, air quotes, accidentally cause harm, right? But why don't our regular M1, hungry hippo macrophages, as I like to think of them, just eat all the cancer up early and kill it and make us all cancer free forever? Because, they're bad cells. Macrophages eat bad junk cells all the time. So why aren't they just getting fat and happy and keeping us cancer free?
That's an excellent question. And we think that one of the major reasons that macrophages at baseline in the tumor microenvironment don't recognize cancer cells as foreign, as bad material to attack, is due to this concept of macrophage immune checkpoints. And so just like there are critical checkpoints that regulate the ability of T cells to recognize cancer cells as foreign and attack those cancer cells, things like the PD-1, PD-L1 interaction or CTLA-4, we believe that there are similar regulatory interactions that control the ability of macrophages to recognize cancer cells as foreign and attack those cancer cells. And the best characterized macrophage immune checkpoint is the CD47 SIRPα interaction. CD47 is a molecule that's highly expressed on the surface of many different types of cancer and also on many normal healthy tissues. And it binds to an inhibitory receptor that's present on the surface of macrophages and other myeloid cells. And this is the SIRPα receptor. When CD47 binds to SIRPα, it sends inhibitory signals to the macrophage that prevent macrophage phagocytosis, prevent macrophage cytokine secretion, and prevent the macrophage from attacking and destroying that cancer cell. And so if you think about this interaction, it's very analogous to the PD-1, PD-L1 interaction, but instead of controlling the adaptive immune system, it's specifically controlling macrophages in the tumor microenvironment.
Does CD47 bind other things? Is that the canonical function of CD47, preventing cells from being attacked by macrophages?
So we think that's probably the major function of the CD47 SIRPα, the CD47 SIRPα interaction controlling myeloid cells in the tumor microenvironment. There have definitely been other ligands of CD47 and other ligands of SIRPα that have been described, but some of them seem to be more context dependent. Some of them seem to be minor ligands. Some of them are restricted to certain cell types. But what's consistent and kind of most predominant seems to be CD47 and SIRPα.
So where's the monoclonal antibody to block CD47 and then, just get in the clinic with the sucker?
Yeah, so so that has been a long journey. So that's a great question. Back in 2008, 2009 timeframe, the initial studies looking at anti-CD47 antibodies coming out from the Weissman lab out at Stanford showed that you could block this interaction with a monoclonal antibody, that when you block this interaction, you lower the kind of activation barrier for a macrophage to recognize the cancer cells as foreign, and attack those cancer cells. Since then, these antibodies, additional therapies have been developed, things that block CD47, things that block SIRPα on the macrophage side, different recombinant proteins that we've made. And consistently, what we see is that the biology is true, the biology is genuine, and that blocking and disrupting the CD47 SIRPα interaction in multiple different preclinical models can show anti-tumor efficacy and can show macrophage activation in vitro, in vivo. These therapies have now moved into testing in clinical trials. So the first under investigation was an anti-CD47 antibody that's now known as magrolimab, that was initially developed by 47 as a spin-out from some of the work from the Weissman lab at Stanford, and has now been acquired by Gilead. They were testing in both solid tumors and hematologic malignancies. The biggest challenge with this antibody has been on-target hematologic toxicity. In particular, it's been known that CD47 is expressed highly on cells in the blood, very highly on red blood cells, and that when you have an anti-CD47 antibody that binds to CD47, blocks the interaction with SIRPα, and has a functional FC on it, something that can activate FC receptors as well on macrophages, that both releases the brakes on the macrophage by blocking the kind of immune checkpoint of CD47, but also steps on the gas by activating FC receptors, and that this causes on-target toxicity to cells in the blood. So that's been one major challenge to CD47 therapies targeting CD47. A second challenge is that there's a large, what we call an antigen sink, where just because there's so much CD47 on cells in the bloodstream, that you have to have a pretty high dose of your anti-CD47 antibody or your CD47 blocking therapy to kind of fill up the sink before you get effective blockade of CD47, presumably in the tumor microenvironment, where you want to activate the macrophages and you want to get an anti-tumor response. And so those are two of the challenges that different groups are trying to overcome right now. The third is kind of this concept of single agent activity versus, so monotherapy activity versus combination strategies. And so from what a lot of the preclinical work on the CD47 SIRPα interaction has shown is that these therapies tend to work best when you have a separate tumor opsonizing antibody that binds to the cancer cells and marks those cancer cells for destruction by the immune system, something that kind of butters up the cancer cell and makes it more palatable to the macrophages. So an antibody like rituximab targeting CD20 on lymphoma cells, or an anti-HER2 antibody targeting HER2-positive breast cancer or other HER2-positive malignancies, an anti-EGFR antibody targeting EGFR-positive cancers. And so it seems like these therapies work best specifically in combination with some of these other FDA-approved antibodies. And so that's where some of the clinical trials that are ongoing right now have been most successful, and where many of the groups are now starting to focus on. So these therapies are under investigation in clinical trials by multiple different groups. And I think hopefully time will tell which approach and which strategy is most effective and which patient population can benefit from them most.
I think that you mentioned this combination therapies. There's so much, not only for CD therapies targeting CD47, but I think in general, there's the problem is, of course, there's so many permutations of different drugs that you can combine. But if you do it in a smart way, there's a lot of potential. I wonder regarding the sync, or is there any way in which you can have like molecules that have double specificity, and maybe you can engineer the CD47 to be a little bit less like the binding less strong, but then you have a second binding to something that is also expressed in your tumor cell, and then that kind of helps the drug or the antibody go persist by the or target the tumor cells preferentially. Is that something also that people are looking into?
Yeah, that's a great question. So a few years ago, we engineered what we called BiMEs, or bispecific macrophage engagers, where one arm would bind to SIRPα and block SIRPα on the macrophage side, and the other arm would bind to the cancer cells. So those look like they work in vitro. They work comparably to the combination of a CD47 blocking therapy separately with a tumor opsonizing antibody. They seem like they work in preclinical in vivo models and mouse models, and haven't yet begun testing in clinical trials. But I think it's a really great approach, and I think we'll see how some of the kind of novel bispecifics start to play out. There are a couple different companies that are now developing bispecifics, mostly focused on targeting CD47 and another antigen. So both antigens, presumably on the cancer cells for the most part, or another immune checkpoint. I think you're absolutely right that because we know CD47 is such a profound inhibitory signal to macrophages, that even if there are obstacles or challenges to some of the first generation strategies for targeting CD47, I'm very confident and very optimistic about some of these next generation strategies, whether it's combining with a CD47 blocking therapy that has a silent FC, looking at bispecific agents, looking at alternative routes of administering these therapies or different kind of engineering strategies. I think it's a matter of time before we kind of crack the code for how to best target this interaction.
That sounds very exciting. And regarding the cancer types, I know that your initial research, at least from your lab, from your work, is what was first on small lung cell cancer, small cell lung cancer. But are there any particular cancer types that seem to be more promising? You also mentioned the hematological malignancies, but where do you think is the best starting point?
That's a good question. So I think definitely what's guided us and guided some of our research that's going on in the lab right now is that the combination of kind of an anti-CD20 antibody for lymphoma with a CD47 blocking therapy is just an extraordinary combination that looks very encouraging in the ongoing clinical trials. Something where kind of there's lots of proof of concept from multiple different groups in clinical investigation in some of the early phase 1 studies. And something where we just see robust synergy in vitro and in vivo and preclinical models. And so that's inspired some of the work that's going on in my lab right now to take a step back and say, can we identify in an unbiased way new antibodies or new antibody combinations that might be very effective for targeting lymphoma cells in stimulating macrophage phagocytosis or stimulating macrophage-mediated destruction. And so we've developed some really cool high throughput assays to evaluate macrophage anti-tumor function in vitro using primary human macrophages and human lymphoma cells. And we've started to screen arrayed libraries of monoclonal antibodies against hundreds of different cell surface targets to identify both the targets that might respond best to opsonizing antibodies or opsonizing therapies on lymphoma cells, as well as which antibodies might be the best antibodies for stimulating macrophage phagocytosis and macrophage anti-tumor functions or anti-lymphoma functions. And so coming back to your prior question, we've started to incorporate all of this knowledge, all of this information from these screening efforts to say, now we've identified a bunch of new targets. Now we've identified a bunch of new antibodies that are very effective at stimulating macrophage phagocytosis. Can we start engineering these things in a rational way to put some of these building blocks together and make novel bispecifics? So we've created a bispecific platform where we've engineered something like 156 novel bispecifics that target either two antigens on the lymphoma cell side or an antigen on lymphoma and a macrophage immune checkpoint molecule. And we've tested them in a high throughput fashion, all side by side, all head to head, to see which antibodies are the most effective at stimulating macrophages to attack the lymphoma cells. And so that's some work that's ongoing in the lab, but something that looks very encouraging. We've identified some really exciting candidate therapeutics that look great in a few different in vitro models, look great in some preclinical models, and hopefully down the line, we'll be able to translate those therapeutics, those candidate therapeutics into testing in patients with lymphoma and testing in investigational studies as well, in clinical trials.
So is biotech spinouts and licensing IP something you anticipate in your career future, or are you more trying to focus on proof of concept, publish it, and then just pass the torch via papers?
I would say everything is something that we're interested in. So I say that the mission statement of my laboratory, the mission statement of the Weiskopf lab is number one, to understand the fundamental biology of how macrophages interact with cancer cells. So to understand some of the molecular mechanisms and what's driving kind of the immunosuppressive function, but also identify things like novel macrophage immune checkpoints that we might be able to target in different therapeutic contexts. And then number two, in parallel, to say, how can we design and develop and engineer new therapies and translate our discoveries to actually make things that benefit patients with cancer or other life-threatening diseases. And so we've had experience developing some novel platforms, developing and identifying new therapeutic targets, developing new therapeutic candidates. And then going back to my time out at Stanford as a graduate student, I've had opportunities to be involved with a few different biotech startups and a few different biotech companies. And so I generated some of the foundational intellectual property for the company 47 that was launched from the Weissman lab to develop the anti-CD47 antibody. I separately co-founded a company, ALX Oncology, that's developing a next generation CD47 antagonist that has a silent FC on it. And then a few years ago, after starting my lab here at the Whitehead Institute, we had a spin out from the lab DemBio in collaboration with Jonathan Weissman's lab here at the Whitehead Institute, as well as Mike Basik's group from Stanford, to create a novel screening platform, a CRISPR-based screening platform to identify novel macrophage immune checkpoints or novel myeloid immune checkpoints, and then develop drugs against some of the new therapeutic targets that we identified. And then more recently was involved with another, as a co-founder of another biotech startup, Solu Therapeutics, that's developing a novel technology to unlock intractable extracellular targets, cell surface targets for cancer and other types of diseases as well. So I've had the opportunity to be involved in a number of different companies, and I really see focusing on the fundamental biology as well as in parallel thinking about ways to translate our discoveries and make new therapies, research that goes hand in hand. And the best way to get those therapies into patients is by partnering with industry and really being able to have an investment to be able to lead those clinical efforts and clinical trials down the line.
I have to say that is super exciting, and I think I really like when we have our guests speaking about exactly that, how they come from these fundamental discoveries and they stay involved in the translation and the uh, trials and the commercialization. And I think a lot of our listeners are also really interested. I think many of them consider this as a career path or something that you can be a bench scientist, but you can also be like an entrepreneur, a uh, agile person making these decisions. For those listeners that might wonder how it is in kind of in practice to be involved with this entrepreneurial endeavors, would you like to maybe just briefly comment on how is it, how you have this discovery, you say you have from your data CD47 is a great target. Can you, in a way, maybe describe how do you come from that to 47, the first company, or your ALX Oncology? I think maybe that's you've been more involved in that company. How is the transition? How does it work?
Yeah, that's a really good question. So it's something where um, I think the more opportunity I've had to be involved, I think, like anything, the greater insight you have into the process and how things can work best. And so I'd say, in thinking about things in the lab right now, absolutely, the number one driver is the science. The number one driver is the biological mechanism, how we can understand which diseases might respond best to certain therapies, which diseases might respond best to certain approaches, what type of platform we can create to kind of develop new drugs or new modalities that might go after some of these kind of genes and signals and hits that we identify. And so that has to be the driver all along. But then also thinking about once we have new targets in hand, once we have things that we think are really exciting, how can we create the best therapeutic candidate, the best molecule to actually leverage the biology that we've learned and really translate those discoveries. And so that is another whole iteration that we're kind of going through constantly in the lab to think about, does it make sense to go after this target with a conventional antibody? What type of FC? How do we want to design this molecule? What type of FC do we want to put on this? Should we be thinking about a CAR T cell for this target? Should we be thinking about an antibody drug conjugate for this target? And then what has the kind of quickest or most efficient route to getting this into patients and actually benefiting patients where there's a large unmet clinical need, where there's a patient population where we think this could be directed to, where this could be a transformative therapy and benefit these patients. And so all of that, I think, is always a conversation between the science, between our intellectual property office here at the Whitehead Institute or at other institutes, the tech transfer offices, conversations with potential investors, venture capital firms who might be interested in some of the work and making those connections, conversations with other scientists or other potential consultants or advisors who might be interested in the work or who might help kind of complete a piece of the puzzle where they have certain expertise and know how to kind of move this type of therapy or modality into the clinic. And I think, it's kind of an integration of all those different aspects, making those connections, having those opportunities to sit down with different people and have discussions, really think about the science and how we can drive it forward and hand it off to other folks who might be able to carry it along the way, I think is kind of the root of how to do things successfully.
So I know I could go on this ankle for a long time, as also being in biotech, but that's not quite this podcast. So we have to shift gears here and kind of go to the fun wrap-up questions. So we're going to play a game of fill in the blank. All right?
All right.
All right. So first up, when I am not conducting research, I am...
I am chasing around my 3 wonderful children. So that, we have my wife and I have 3 young kids, ages 3, 5, and 7. Our 2 oldest are girls, our youngest is a boy. They're wonderful and so much fun to hang out with and occasionally talk science with. They're all macrophage enthusiasts, and they have tons of energy and keep us busy. But I love doing kind of all the dad activities. So we're always going to museums. Here in Boston, we love the Museum of Science. We love the aquarium. I coach soccer for our 2 girls in kind of their town youth league. I love taking them mini golfing, love taking them skiing. So we have a lot of fun, but they definitely keep us busy.
All right. And if you could have one superpower, what would it be?
I think as a physician, the superpower I would desire most would be the ability to heal, the ability to heal others, and something where I feel like the most satisfying thing in medicine is to be able to give a therapy where a patient instantly gets better and has a remarkable kind of change in their disease course or trajectory. But often we don't have those types of therapies for many types of diseases. And just, a superpower I wish I had would be the ability to heal, where we can overcome some of those obstacles instantly and really be able to change the direction of patients' lives.
All right. And what do you need every morning to get going?
Definitely coffee.
Those were all very original replies. Maybe the last one, not so much, but
Yeah.
but was very, very. Thank you so much for sharing with us and with our listeners.
Are you on social media anywhere? Are you looking for grad students or postdocs that you want to post an ad for here at the end of the wrap up here?
Always interested in recruiting new lab members. So research technicians, grad students, postdocs, anybody that's looking for an opportunity in macrophage immunotherapy, developing novel immunotherapies, antibody therapies for cancer, other diseases. Of course, feel free to reach out. Would love to talk.
Perfect.
Wonderful. Well, thank you so much. We had today in the show Dr. Kipp Weiskopf from the Whitehead Institute talking to us about cancer therapies and new why macrophages are actually pretty cool.
That's right. Thanks so much for having me on the show.
Thank you for being here.
So that brings us to the end of our show. Don't forget to subscribe to our newsletter at www.immunologypodcast.com to get the show notes, including an episode summary and links to all of the interview and Roundup Papers. You can also reach out to us on X at Immunopodcast or via email at info@immunologypodcast.com if you had feedback or you would like to suggest a guest. See you next time.